Hi guys, this is Dr. Rajeshwar from YR Pharma Tube. In the previous video, we understood the glycolytic pathway. If you did not watch the video, hit the I button on top right of this video to watch it. To watch the other topics of biochemistry and other subjects of pharmaceutical sciences, click on the links given right below this video. In this lesson, we shall discuss on the energetics, significance, regulation and fate of glycolysis. Energetics of glycolysis. It is understood from the previous lesson that each molecule of glucose yields 2 molecules of pyruvate via glyceraldehyde 3-phosphate. The total inputs and outputs of all the 10 glycolytic reactions may be written as shown in the equation here, where glucose utilizes 2 molecules of ATP, 2 monophosphates, 2 NAD+, 2 protons or H+, and 4 ADPs to give 2 molecules of pyruvate, 2 protons, 4 ADPs, 2 molecules of water, 2 NADH, 2 protons and 2 ADPs. On cancelling the common terms, we get the net equation for the transformation of glucose into pyruvate as shown in equation 2. Thus, 3 things happen simultaneously in glycolysis. Number 1, glucose is oxidized to pyruvate. Number 2, NAD+ is reduced to pyruvate. And number 3, adenosine diphosphate is phosphorylated to form adenosine triphosphate. It is to note that there can be no glycolytic pathway without these 3 events, which means that NAD+, ADP and monophosphate as well as glucose must be present. During anaerobic condition, that is oxygen deficient or without the presence of oxygen, when 1 molecule of glucose is converted to 2 molecules of lactate, there is a net yield of 2 molecules of ATP. 4 molecules of ATP synthesized by the 2 substrate level phosphorylations. For clarification, please refer the previous video. But molecules of ATP are used in the step 1 and 3, hence the net yield is only 2 ATPs. The whole reaction can be summarized as glucose plus 2 molecules of monophosphate and 2 molecules of ADP, which gives rise to the formation of 2 lactate molecules plus 2 ATPs. But when oxygen is plenty, that is in the presence of oxygen or in aerobic conditions, the 2 NADH molecules generated in the glyceraldehyde 3-phosphate dehydrogenase step can enter the mitochondrial electron transport chain for complete oxidation. As each NADH provides 2.5 ATPs, this reaction generates 2.5 into 2 is equal to 5 ATPs. Thus, when oxygen is available, the net gain of energy from the glycolysis pathway is 7 ATPs. Hence, the ATP yield from glycolysis is different in anaerobic and aerobic conditions. In aerobic conditions, pyruvate is converted to acetyl-CoA, which enters the TCA cycle for complete oxidation. TCA cycle or the reactions involved in the TCA pathway will be discussed in the later videos. Complete oxidation of glucose through glycolysis plus a citric acid cycle will yield a net 32 ATPs. Significance or importance of glycolysis. Glycolysis is the principal route for glucose metabolism for the production of ATP molecules. Glycolysis is the only pathway that is taking place in all the cells of the body and provides energy to the cells. It is the only source of energy in erythrocytes. In strenuous exercise, when muscle tissue does not contain enough oxygen, aerobic glycolysis forms the major source of energy for muscles. Since heart is mainly aerobic organ, myocardial ischemia decreases glycolytic ability of cardiac muscle. As a result, energy or ATP production in heart is affected. Deficiency of enzymes such as pyruvate kinase of erythrocyte glycolysis causes hemolytic anemia. This is because erythrocytes get their energy from glycolysis. Deficiency of muscle phosphofructokinase enzyme causes decreased muscular performance and fatigue. Dietary fructose and galactose are also metabolized by glycolytic pathway. Glycolysis has amphibolic role also. It provides precursors for the formation of lipids and amino acids. For example, pyruvate is converted to alanine by transamination and dihydroxyacetone phosphate serves as a precursor for triglyceride formation. Two glycolytic intermediates, pyruvate and glyceraldehyde 3-phosphate, are used for the synthesis of cholesterol, thiamine, and pyridoxine in tuberculosis, malaria, and gastritis causing organisms. Glycolysis is the major energy source for rapidly growing malarial parasite in RBCs. Lactate is the end product of glycolysis in malarial parasite. Lactate dehydrogenase of parasite is different from human enzyme. Unlike human, lactate dehydrogenase parasite enzyme is not subjected to inhibition by substrate pyruvate. This allows rapid formation of lactate from pyruvate and fast energy production. In brain tumors, lactate production is 10 times more. Regulation of glycolysis. Glycolysis is regulated at 3 different steps which are irreversible reactions. These 3 reactions are catalyzed by number 1 glucokinase and hexokinase, number 2 phosphofructokinase, and number 3 pyruvate kinase. Hexokinase and glucokinase. Hexokinase is an allosteric enzyme that catalyzes the conversion of glucose to glucose 6-phosphate and thus initiates the glycolytic pathway. This enzyme is inhibited by a high concentration of glucose 6-phosphate, the product of the reaction it catalyzes. Thus, the phosphorylation of glucose is a reaction which is regulated by the feedback inhibition, that is hexokinase by glucose 6-phosphate, and activated by insulin, that is glucokinase is induced by insulin. Glucokinase is active mainly in liver and has a high Km value for glucose and low affinity. Km is the Michaelis constant value of the enzyme, which is an inverse measure of affinity. For practical purposes, Km is the concentration of substrate which permits the enzyme to achieve half Vmax, where Vmax is the maximum reaction velocity at which all enzymes become saturated with the substrate. Hence, glucokinase can act only when there is adequate glucose supply so that excess can be stored. Hexokinase with low Km value and high affinity can phosphorylate glucose even at lower concentrations so that glucose is made available to the brain, cardiac muscle, and skeletal muscle. Glucokinase can act only when there is a plenty of glucose. Thus, when the supply of glucose is limited, that is reduced glucose supply, glucose is made available to brain and muscles. Liver glucokinase is an inducible enzyme that increases its synthesis in response to insulin and decreases in response to glucagon. Phosphofructokinase. A second and very important regulatory point for glycolysis is the conversion of fructose 6-phosphate into fructose 1,6-bisphosphate, which is catalyzed by the enzyme phosphofructokinase. Once glucose is converted to fructose 1,6-bisphosphate, its carbon skeleton must continue through the glycolytic pathway. The phosphorylation of fructose 6-phosphate is a committed step that will occur as long as the enzyme is active. As an allosteric enzyme, phosphofructokinase is inhibited by high concentrations of ATP and citrate, and it is activated by high concentrations of ADP and AMP. A high ATP concentration, which is characteristic of a state of low energy consumption, thus stops glycolysis at the fructose 6-phosphate stage. This stoppage also causes increases in glucose 6-phosphate stores because glucose 6-phosphate is in equilibrium with fructose 6-phosphate. Phosphofructokinase is an inducible enzyme that increases its synthesis in response to insulin and decreases in response to glucagon. Pyruvate kinase. The third regulatory point is the last step, that is step 10 of glycolysis, that is the conversion of phosphoenol pyruvate to pyruvate. This reaction is catalyzed by the enzyme pyruvate kinase, which is an irreversible step, and thus it is a regulatory enzyme of glycolysis. This pyruvate kinase enzyme is also an allosteric enzyme that is inhibited by high concentrations of ATP. When energy is plenty in the cell, glycolysis is inhibited. It is to note that insulin increases its activity whereas glucagon inhibits. Also, pyruvate kinase is inactive in the phosphorylated state. Now coming to the fate of pyruvate. The formation of pyruvate from phosphoenol pyruvate is the last step of glycolysis. We should note that the pyruvate produced from the glucose is not the end product of glucose metabolism. The pyruvate enters the other pathways that continue to extract energy. Further metabolism of pyruvate typically takes one of the 5 routes as shown in the figure here. Number 1. Pyruvate can be converted to acetyl-CoA, and this acetyl-CoA can be used in a number of metabolic pathways. In one of the important pathways such as the citric acid cycle, pyruvate is completely oxidized to carbon dioxide. This is a route that operates efficiently in the presence of oxygen. Number 2. Pyruvate can be carboxylated to produce oxaloacetate. Oxaloacetate is one of the citric acid cycle intermediates, but it is also an intermediate in the synthesis of glucose. Number 3. In some species, pyruvate can be reduced to ethanol, which is then excreted from the cells. This reaction normally takes place under anaerobic conditions where entry of acetyl-CoA into the citric acid cycle is unfavorable. Number 4. In some species, pyruvate can be reduced to lactate. Lactate can be transported to cells that convert it back to pyruvate for entry into one of the other pathways. This is also an anaerobic pathway. And number 5. In all species, pyruvate can be converted to alanine. Oxidation of pyruvate to acetyl-CoA. In glycolysis, 2 ATP are generated when one glucose molecule is converted to 2 pyruvate molecules. However, much more energy is obtained from glucose when oxygen levels are high in the cells. Under aerobic conditions, pyruvate moves from the cytosol into the mitochondria to be oxidized further. In a complex reaction, pyruvate is oxidized and a carbon atom is removed from the pyruvate as carbon dioxide by the action of pyruvate dehydrogenase enzyme. The coenzyme NAD+ is reduced during the oxidation reaction. The resulting 2-carbon acetyl compound is attached to CoA, producing acetyl-CoA, an important intermediate in many metabolic pathways. Reduction of pyruvate to lactate. When we engage in strenuous exercise, the oxygen stored in our muscle cells is quickly depleted. Under anaerobic conditions, pyruvate remains in the cytosol where it is reduced to lactate. The product NAD+ is used to oxidize glyceraldehyde 3-phosphate in the glycolysis pathway, which produces a small but needed amount of ATP. The accumulation of lactate and the corresponding drop in pH causes the muscles to tire and become sore. After exercise, a person continues to breathe rapidly to repay the oxygen debt incurred during exercise. Most of the lactate is transported to the liver where it is converted back into pyruvate. Bacteria also convert pyruvate to lactate under anaerobic conditions. In the preparation of kimchi and sauerkraut, cabbage is converted with salt brine. The glucose obtained from the starches in the cabbage is converted to lactate. The acid environment acts as a preservative that prevents the growth of other bacteria. The pickling of olives and cucumbers gives similar products. When cultures of bacteria that produce lactate are added to the milk, the acid denatures the milk proteins to give sour cream and yogurt. Reduction of pyruvate to ethanol. Some microorganisms, particularly yeast, convert sugars to ethanol under anaerobic conditions by a process called fermentation. After pyruvate is formed in glycolysis, a carbon atom is removed in the form of carbon dioxide. The process is known as decarboxylation. The NAD plus for continued glycolysis is regenerated when the resulting ethanol is reduced to ethanol. The type of carbohydrate used in the reaction determines the taste associated with a particular alcoholic beverage. Beer is made from the fermentation of barley malt, wine, and champagne from the sugars in grapes, vodka from potatoes or grain, sake from rice, and whiskies from corn or rye. Fermentation produces solutions of about 15% alcohol by volume. At this concentration, the alcohol kills the yeast and fermentation stops. This is the list of references followed for the lesson. That's all in this video, the energetics, significance, regulation and fate of glycolysis. In the next lesson, we will discuss about the citric acid cycle. Till then, never stop learning and never stop watching my videos. Thank you for watching this video.